In this lecture, we're going to discuss the viral replication cycle, the viral host interaction at the cellular level, antiviral drugs, and the pathogenesis of viral disease. Before we start the viral replicated cycle, we must remember that the viruses are obligate intracellular parasites, which replicate only in living cells. The replication cycle of the virus starts by attachment of the virus to the specific host cell receptor, followed by entry or penetration of the virus to the host cell, followed by uncoating, as the virus when it enters the cell is with its capsid. So the genome to be free, the virus must be uncoated to complete the replication. Now the virus is free in the cytoplasm of the host cell. So this genome of the virus or nucleic acid will form, or from it we will form the protein and the nucleic acids of the new viruses. Forming of the protein starts by transcription and formation of the messenger RNA and translation on the ribosomes. And formation of nucleic acids of the new viruses is via the replication of this genome. By forming the proteins and nucleic acids of the new viruses, the second step will be assembly, which is getting together both the nucleic acid and the proteins. Now the mature viruses are formed and become released from the infected host cell. This is simply how the virus replicates in the host cell. In some, we will discuss the attachment, which is the first step in the replicated cycle. Attachment occurs to a specific host cell receptor. Example, the human immunodeficiency virus attached to the CD4 molecules on the T lymphocytes. This is an important factor involved in viral tropism, which will be discussed in the pathogenesis of viral disease. After attachment to the host cell, the virus gains entry or penetration to the cell. This penetration occurs by either fusion of the virus with the cell membrane of the host cell with the release of nucleocapsid in the host cytoplasm, or by endocytosis, where the cell membrane invaginates around the attached virus. After entry of the virus in the host cell, either by fusion or by endocytosis, comes the step of uncoating. The virus enters with its coat, and the virus needs the genome to be free so as to be transcribed and to replicate by itself. So here is the step of the uncoating, where the protein coat is removed mainly by the host cell enzymes in the lysozymes. Being free, this genome will act on two important synthesis processes. First, the protein synthesis, second, the genome synthesis. You know, the virus is composed of the genome surrounded by the capsid. So these new viruses need new genome and new capsid. How is this capsid formed and how is this new nucleic acid formed? Is through this free genome. First, as regards the protein synthesis. First, transcription. Transcription, this genome is transcribed with the production of the messenger RNA using the viral genome as a template. Then this messenger RNA will be translated on the host ribosome to form the specific viral proteins. As regards the nucleic acid synthesis, the template is the parental genome. And if this genome is single-stranded, then first it forms a complementary strand, which then acts as a template for formation of other genomes. Here again, the viral genome, the free viral genome in the host cell is important for the gene expression, transcription and translation, and formation of the viral proteins. It's important for formation or replication and forming copies of the viral genome. That's why, if you remember, we have mentioned that the genome is responsible for infectivity. As regards the assembly process, which is getting together both the genome, or the newly formed genome, and the protein coat or capsid to form the mature viral particles. Then the virus becomes released from the host cell by lysis or rupture of the host cell, which usually occurs in the non-enveloped viruses, or by budding through the cell membrane as in the enveloped viruses. As regards the antiviral chemotherapy, here the antivirals have certain limitations because, as we have mentioned, that the virus can't replicate except inside the living host cells. So these antivirus must selectively inhibit the viral function without affecting the functions of the host cell. Also, during the incubation period of the disease, many rounds of virus replication have occurred. So the virus have been spread before even the symptoms appear, which make the drug relatively ineffective. Mechanism of action of antiviral drugs. Here the antiviral drugs depend on inhibiting steps in the replication cycle. If you remember, the antimicrobial, antibacterial agents target certain structures, whether cell wall, ribosomes, and so on. But in the case of the antiviral drugs, they target steps in the replication cycle. There are drugs inhibiting penetrating and uncoating, inhibiting nucleic acid synthesis or an integrase enzyme, or the protein synthesis or protease or inhibition of the release. Towards inhibition of the penetration, there are drugs which inhibit the fusion, inhibits the penetration by fusion. Also, there are drugs which inhibit the uncoating. If we inhibit the uncoating, the genome will not be free, so the virus can't complete the replication. They are better to be administered prophylactically to have a significant protective effect. Also, inhibition of nucleic acid synthesis. There are DNA polymerase inhibitors which inhibit the formation of the DNA of the virus, and also RNA polymerase inhibitors which inhibits the formation of the viral RNA. Reverse transcriptase inhibitors are important inhibitors for HIV or the human immunodeficiency virus replication, because these viruses depends on these enzymes. These enzymes are RNA and need to be transformed into DNA via these enzymes to complete their replication cycle. So if this enzyme is inhibited, so the HIV is inhibited or prevents its replication. And there are drugs interfering with the messenger RNA synthesis. Also, there's an enzyme in the human immunodeficiency virus which is responsible for integration of the DNA formed by the reverse transcriptase, and this DNA becomes incorporated inside the host DNA by the integrase enzyme. So this integrase enzyme is responsible for integration of the newly formed DNA into the host chromosome. There are drugs inhibiting these enzymes, thus inhibiting the human immunodeficiency virus. Also, there is inhibition of the protein synthesis, the interferon. There are recombinant forms of alpha interferon which are used in chronic hepatitis B and C. These interferons induce the production of enzymes that inhibit protein synthesis. As regards inhibition of cleavage of precursor polypeptides, we have viruses as the human immunodeficiency virus, in which the proteins after translation are complex formed, and they need the viral protease to break down these proteins to be used as structural and non-structural proteins to form the enzymes and capsids and so on. So if this enzyme, this protease enzyme responsible for breaking down these polypeptides is inhibited, then it will inhibit the production of mature viruses. The mature viruses need the genome plus the proteins. So this is another mechanism by which drugs can inhibit the replication of the virus. The last example for antiviral drugs is the inhibition of the release of the virus by neuraminidase inhibitors. Neuraminidase enzyme is an important enzyme in the influenza viruses. important for the release of the virus from the host cell. When there is an inhibition of the release of the virus, then there will be limitation of the spread of the virus from one cell to another. So, again, these antiviral drugs acted on steps of the replication cycle of the virus. As regards the pathogenesis of viral disease, how can this virus cause disease? First, the virus must enter the host, whether by inhalation, ingestion, sexual contact, vertically. Vertically means from the mother to her fetus, whether transplacentally or during passage in the birth canal or by milk. Some viruses also can enter the body through the skin abrasions, injections, or bite of an animal or an arthropod vector. After the virus enters the body, it replicates at the site of entry. Some viruses cause disease at the site of entry, local infection at the site of entry, as the influenza virus causes respiratory tract infection and the rotavirus causes GIT infection. But other viruses spread to their target organs as the enteroviruses. These enteroviruses are transmitted fecal-orally, enters by ingestion. Then, after passage in the intestine, they are absorbed to the blood, replicating the patch, then absorbed into blood, causing viremia, and from the blood they spread to their target organs. So spread of the virus might be by blood or lymphatics, and it also can be transmitted via the neurons, as in case of rabies. Mechanism of cell tropism. What is cell tropism? Cell tropism means the virus affinity for specific body tissue. Why the virus prefer specific body tissue? First mechanism is the presence of specific cell surface receptor for the virus. You remember when we mentioned the attachment, and the attachment to specific, we named it specific cell receptor. Each virus has a specific cell receptor, as the HIV has its receptor, the CD4 molecules. So this is one of the mechanisms of cell tropism. Second mechanism is the local temperature, pH, and oxygen tension. For example, the rhinoviruses, and you know rhino comes from the nose. These viruses multiply at low temperature and high oxygen tension. That's why these rhinoviruses cause common cold, affecting only the upper respiratory tract, because it replicates mainly in the nose, where there is low temperature and high oxygen tension. A mechanism for cell tropism is the proteolytic enzymes. Some viruses need certain proteolytic enzymes to be infectious. For example, we have paramyxoviruses, which have certain glycoproteins on its surface, which is responsible for attachment to the host cell. These glycoproteins are not active except after certain modification. This modification is performed by the proteolytic enzymes, which are present in certain sites, which in case in the paramexo, they are present, these proteolytic enzymes are present in the respiratory tract. So, infection starts in the respiratory tract because of the presence of these proteolytic enzymes, which modifies the glycoproteins on the surface of the virus. Okay, the virus entered the cell and caused certain changes in this cell. Might cause the cell death by the cytopathic effect of the virus on the cell. So the cell can die because of the cytopathic effect of the virus, but it also can die by the effect of the immune response. As you know, the viral antigen will be presented by the MHC class 1 on the surface of the viral infected cell, and these viral infected cells will be recognized by the cytotoxic T lymphocytes and the natural killer cells, which will cause the death of this infected cell. So the cell death will be by the virus itself or by the immune response to the viral infected cell. This cause lesional dysfunction in the organs, which will be in the form of symptoms and signs. Most tissue will rapidly regenerate, but some do not, as the brain or the CNS. There is no regeneration in these tissue. Another effect of the virus on the host cell is cell transformation, where the cell here is not killed, but it will change into a malignant form. As regards the host immune response, we have the humoral immune response, antibody formation, cell-mediated immune response, interference, and other host defense factors. As regards the fate of the virus, usually there is complete viral clearance and complete resolution. However, in some cases, the virus remains persistent inside the host cell. We have three forms of persistence: chronic infection, latent infection, and slow infection. As regards chronic infection, in this case, the virus is continuously detected with mild or no clinical symptoms. Even if there is no symptoms, the virus can be detected in this host, as in case of hepatitis B virus. But in the other form, the latent infection, the virus persists in an occult form inside the infected cell. The virus is dormant, and it is not detected. And this latent virus can be reactivated later when there is lowering in the immune response. Can be reactivated and produce the disease again. So the first form was chronic infection, and the virus is detected. The second, latent infection, but the virus is not detected. And the third form is slow virus infections. In this infection, the incubation period is long, even years. Example, multifocal leukoencephalopathy, which is caused by the JC virus. So these are the three examples or forms of persistence of the virus inside the host cell. Finally, the virus becomes shed into the environment, and this is when the person becomes infectious, because he spreads the virus to another person. Usually, it is shed from the body surfaces involved in the entry. For example, fecal-oral route, then it will be present in the stool. For example, respiratory route, it will be present in the respiratory secretions, and so on. Humans may be a dead-end host, and shedding doesn't occur, which means that the person is not infectious, as in rabies, meaning that this person will not transmit the virus to another person. Types of viral disease. There are systemic diseases or localized disease. In the systemic disease, the virus spreads and invades many tissues. Localized disease, from its name, it invades only the tissue adjacent to the site of entry. Incubation period in case of systemic is long, and the localized is short. As regards the immunity, the systemic produce long-lasting immunity, while the localized transient immunity.